Okay, so today's lecture is going to be about the process of mitosis, focusing on cell division in animals. And this, of course, is part of the cell cycle, and most cells go through this at some point. Most go through it regularly, where they copy their DNA during a different part of the cell cycle, and then have to figure out a way how to divide that DNA equally between the two daughter cells, and that is the process of mitosis. So mitosis is separate from cell division, but is definitely part of that whole process. So how is our DNA organized? We talked a little bit earlier about how eukaryote DNA is linear, right? And prokaryote DNA is circular, and that's going to play a bigger part later on when we talk about DNA replication. But for now, we're strictly speaking going to talk about the part of the process where the DNA has already been copied. It's already been replicated, and now we're getting ready to actually divide our genetic material in half so that we can or separate those replicated chromosomes so that we can accomplish cell division. So this is happening prior to this process, right? We've copied all of our DNA, and that means that every chromosome that we have essentially exists as a pair of what we call sister chromatids. And we call them sister chromatids because they are genetically identical to each other, right? We know that we get one chromosome, right? One of each type of chromosome from our parents, right? So prior to cell division, prior to this mitosis and then cell division process, our cells are going to replicate that DNA. So that means that they're going to take the chromosome, say chromosome number 2, that I got from my mom, and they're going to copy it. And then I'm going to have a chromosome that is made up of 2 sister chromatids, and it's going to look like this picture here. And then they're going to take the same one, that's the copy of number 2 that I got from my dad, also copy that. And then that chromosome is made up of 2 sister chromatids. And part of the issue, of course, is when figuring out whether I have, whether I'm haploid, meaning I only have 1 copy, or I'm diploid, meaning I have 2 copies. And for most of our lives, we are diploid. Figuring that out and what happens after DNA synthesis that's confusing is, when does a sister chromatid become a chromosome? And we'll talk about that. But an easy way to figure out kind of how many chromosomes you have is to count the centromeres. And the centromere is basically the location where the sister chromatids are attached. So we can see, though, let's see if I can get this to work. Right here, we have a centrosome, here's a chromosome, right? I haven't copied it yet. And here is the centromere, right? Here, that little red line, right? So when I copy the DNA, now I have 2 identical sister chromatids, but guess what? I still have only 1 centromere. So I'm still diploid, right? I'm not tetraploid at this point. I'm still diploid. I only have 2 chromosomes because this particular chromosome is made up of 2 sister chromatids, right? So that equals one chromosome. And then at some point, of course, we're going to separate the sister chromatids, and they're going to go into 2 different cells, right? And then, okay, now I've got a centromere there, and I've got a centromere there, and now I have 2 separate chromosomes. I know. It's where 1 becomes 2. It's sort of the reverse of that Spice Girls song. Which you guys probably have no idea what I'm talking about. But anyway, we don't have 2 becoming 1. We have 1 becoming 2 here eventually. And that's going to be part of this process that we're going to walk through here today. Okay, so As I mentioned, right, mitosis is separate process from cell division, right? But it is part of that process. So mitosis is essentially the dividing of the genetic material, and that's immediately followed by the dividing of the entire cell, right, from one cell into two cells. And so typically you have mitosis as well as meiosis, which we will be talking about in class, and you guys have covered on the SymBio. So it goes through the same set of phases. So we essentially have 5 phases to both mitosis and one part of meiosis, right? Because we know meiosis is a 2-part process. And there's prophase. We have pro-metaphase, just to keep it interesting, metaphase, anaphase, and then, of course, telophase. So prophase being, this is when the DNA is condensing down. Typically, DNA is, well, the DNA and the chromatin's condensing. We're going to talk about what the heck chromatin is here in just a minute. But chromatin condenses down and forms, basically when we look under a microscope, we can see the chromosomes. And so at the end of prophase, you can see the chromosomes inside the nucleus. You have, and the nuclear wall, the nuclear membrane also dissolves. Prometaphase, things start getting connected and moving with the other parts of the cytoskeleton getting ready to separate. Metaphase, our chromosomes line up along this sort of invisible line in the cell. Anaphase is the actual separation, and then telophase is essentially the reverse of prophase. So by the time telophase is almost complete, the process of cytokinesis is well underway. And cytokinesis is actually the process of cell division. And animal cells and plant cells handle this differently, but the overall idea is generally the same, that you are separating both the genetic material as well as the cytoplasm and everything, all the organelles and everything that you've already duplicated into 2 identical daughter cells. So the whole goal of mitosis is to end up with identical daughter cells that have everything that they need to do their job, and they're exactly the same as the cell that they came from. Okay, so as I mentioned, in prophase, right, the chromatin starts to condense, and chromatin is basically how do I describe that? Our DNA is, of course, the double-stranded, the double helix, right? We've got nucleic acid, right? So deoxyribonucleic acids, and we have the monomers or the nucleotides, and we've linked them together. So you guys know all about the structure of DNA itself. Our DNA is bound to protein complexes called histones. And so the histone plus the DNA is chromatin. Now, typically chromatin is really a very loose sort of mass inside the nucleus. It's really kind of a mess in some ways. And part of the process of the chromatin condensing is basically all of those molecules kind of packed. They pack really tightly together to the point that you can see the actual chromosomes themselves under the microscope in this part of both the phases of mitosis, but also meiosis. So we're starting to pack that chromatin down, and a large reason behind that is it's a whole lot easier to move it when it's packed tightly. Think about sort of like packing a suitcase, right? If you're going on a trip, it's really hard to keep track of all of your items if you just sort of randomly stuff them, say, in, I don't know, garbage bags or something like that, or you're trying to hold them in your arms, or a laundry hamper, right? It's a lot easier if you take all of your important stuff and you pack it in a suitcase. You basically squish it down, and again, if it doesn't want to go, you kind of have to make it go. You might have to shove it in there, pack it really tightly. Now all your important stuff is in one box. It's in one location, and that makes it really easy to pick it up and move it, and it makes it easier to keep track of. You're less likely to lose something, right? If it's packed in a suitcase versus you're just, carrying it around in a big floppy lump in your arms or whatever, and you're dropping socks down the hall, right? So kind of a big mess. So that's sort of the reason behind condensing the chromatin in the first place. And we have to condense it down before we can move things around. If we try to do it without doing that, we may end up with broken chromosomes, and sometimes that happens anyway, actually. But for the most part, we want to make sure that we are packing all our DNA down, and then we're going to move it to the daughter cells. So also as part of prophase, the mitotic spindle starts to form. And this is basically made up of those key cytoskeleton components that we were talking about earlier when we talked about the parts of the cell, right? And so you have essentially an anchor to this. These are the asters here, which are also known as centrosomes, which again is confusing because we have centromeres, which centromeres are holding the chromosomes, the centrosomes are centromeres. I'll get it right eventually. Centromeres are holding the sister chromatids together. The centrosomes are these basically the asters contain the centrosomes. They're clusters of microtubules whose job it is going to be to anchor themselves to the cell membrane on one end, and the chromosomes themselves, in particular close to the centromere, but the chromosomes themselves during division, and basically pull the chromosomes apart. So the microtubules actually grow out from this area and form kind of something that looks sort of like a football-shaped apparatus that we call the spindle. Okay, so refer to the spindle as this kind of football-shaped object, I guess, that's part of the mitosis process and certainly part of the cytokinesis process. So the spindle is made up of a number of different parts. So before this all begins, right, the centrosome actually replicates itself. So a centrosome is basically a region in the cell where you're organizing the microtubules to form the spindle. So you have two centrioles within each centrosome. A centriole is basically this wonderfully delicately constructed star-shaped structure that basically serves as an anchor point for the microtubules that are going to grow out from that, from the centriole, either towards the plasma membrane on one side or towards the chromosomes on the other side. And so you actually have two centrioles per centrosome, right? And so each centriole is going to be involved in either anchoring the microtubules to the plasma membrane or growing out towards the chromosomes, which are going to line up in the middle of the cell. So you can't actually accomplish this whole thing with only one centrosome and two centrioles. So you actually have to copy them first, which has occurred before this, and then in the beginning of prophase, we were seeing as the chromatin is condensing, the centrosomes move, and the centrosomes are then starting to migrate to opposite ends of the cell, right? So each centrosome has got two centrioles, and then from those centrioles, you get what's called the aster, right? And this is just basically this array, this star-shaped array of microtubules that extends out. And one end is going to anchor with the chromosomes, and the other end is going to anchor against the other centriole with a shorter aster is going to anchor against the side of the plasma membrane, and we're going to work together to pull the chromosomes apart as part of this. So basically, the centrosome is the region that has all of the components that are needed to build the microtubules and to build the centrioles, and the centrioles themselves are basically the anchor points. This particular sort of star-shaped structure, and then the aster is basically a term for the microtubules that are growing out from the centriole to meet the chromosomes. So the spindle ends up being basically all those things together. So it's the centrosomes, which includes each centrosome has 2 centrioles, right? It's the microtubules that all of that stuff is made of. And then the asters, which is basically just the arrangement of the microtubules. So the microtubules are the building blocks, and they're the things that are actually doing the work. And then how they're organized, they're organized into asters, which are growing out from the centrioles. And then the centrosome is the region that contains basically all the proteins and everything that are needed to build this microtubule network. And this all starts to form during prophase. Okay, so here you can actually see much more clearly now, right, we're in pro-metaphase. Our chromatin has condensed, and we can actually make out our, if I can get my little doodad to work here, my little pointer. You can actually make out, right here, I've got a pair of sister chromatids to form one chromosome here. I'll make a little dot, or a little, here's the centromere of one pair of sister chromatids, so that's one chromosome, right? I've got another chromosome here, I've got another chromosome here, and so on, right? Here I've got my centrosome is this thing here, right? My centrosome is made up of 2 centrioles, right? One's going to be anchoring the microtubules that are growing towards the chromosomes, and the other is anchoring against, as you can see how the microtubules are growing out, anchoring into the plasma membrane of the cell, and basically going to hold on and act as an anchor point for the cytokinesis process, which is coming much later as part of this. So we have microtubules that are involved in sort of anchoring with each other. So we have microtubules that are coming out and growing out from one centriole. This is again part of the aster, and they're actually going to meet up with microtubules that are growing out from the other centriole to help form this spindle structure, but also basically help organize this whole process. So during pro-metaphase, as we can see, the nuclear membrane has dissolved. So we no longer have basically a double plasma membrane. That's basically broken down into pieces. And the pieces are so small we can't really see them. And we have dissolved the membrane around that basically was packing our DNA. And so now our DNA is sort of free form with the rest of the cytosol, really, and the whole rest of the cell. We no longer have a boundary between the cytosol and the nucleus, the contents of the nucleus. And so as the microtubules are growing out from our centrioles, right, some of those are going to attach to a part of the centromere, again, just to make it really confusing, right? So we can see that here, this kind of pinched area. This is the centromere, and I've got basically protein anchors here, and my microtubule is essentially going to attach to those, almost like a tow hitch or something like that on a car. And the microtubules attach to what's called the kinetochore, and again specific to the centromere, right, not centrosome. So we're on the centromeres. Basically, the kinetochore is where the microtubules attach to the chromosomes on the centromere, and that's basically where they're going to start pulling the chromosomes apart. So in pro-metaphase, you have the spindle is pretty well fully formed. the nuclear membrane has dissolved, and now you have the microtubules are anchored onto the chromosomes via the kinetochores, right? But you notice that our chromosomes, right, which are made up of two sister chromatids, right, still haven't lined up yet on the what's known as the metaphase plate. Okay, so metaphase, this is kind of the fun part. We have this weirdness. So your chromosomes now are lining up at sort of a midway point in the middle of the cell, right? And you have, right, you can see the spindle fibers, which are basically those microtubules coming out from our centrioles here, right? The centrioles and the centrosome are sort of making up the aster here on one end, and we've got another one on the other end. And so we have some microtubules that are, again, anchoring in the plasma membrane, and others that are attached either to other microtubules or that are attached to the sister chromatids, right, at the centriole. And these are the kinetochores, right, or sort of in a darker kind of black color, so you can see where they've attached. And again, here's a scanning electron microscope. So you have the centromere, and you have your kinetochores are here, right, and that's where the spindle fibers are, which are the microtubules, right. That's where those are being attached, and it's going to take that pair of sister chromatids that are sort of being held together at the centromere and start pulling them apart in the next phase. At the moment, our chromosomes are just lining up in the middle, and they line up basically, I don't want to say vertically, but basically along one particular, what we call the metaphase plate, right? This is sort of an invisible plane in the middle of the field. And they don't line up next to each other, they line up adjacent, right? So they're basically forming a long line or a long string of pairs of sister chromatids, right, which are each themselves a single chromosome, just like you can see here on this, again, on this microscope picture. Or you can see that the chromosomes are starting to take their position on the metaphase plate. And the whole point of this, right, is that they line up, and again, it doesn't really matter which chromosome goes first, right, or left to right or whatever, but they're going to line up such that when they are pulled apart, right, each of the daughter cells is going to get one of those sister chromatids, right, which now each has its own centromere, and we're going to call it its own chromosome. But anyway, we're lining up here in metaphase and getting ready for the next part of the process. Okay, anaphase, this is the action part, right? So I always think of like ana, action, right? A for action. So at this point, right, the chromosomes are lined up, and now they're starting to be pulled apart by the microtubules, right, that are attached to the kinetochores, right? You can see the little black kinetochores here and here, right? And now I'm going to make them kind of red, as I will like mark them a little bit here. I've got another one here, right? And that's, right, the kinetochores are attached to the centromeres, and then on one end, right? And they're attached via those microtubules to the centrioles, right, which are together here in the centrosome, which again forms the aster part of the spindle, right, which is including all these microtubule fibers as well. And so I basically, now I have my centrosome on this end, and I have a centrosome on this end, and of course the centrioles are within them are anchored to the plasma membrane. Now the microtubules actually start, oops, there we go. They actually start separating the chromosomes and moving them, and basically the microtubules are getting shorter. They're essentially breaking down at the centriole end, and they're pulling as a consequence. They're moving now the separated sister chromatids, right, which are now fully chromosomes on their own, right, because they each have a centromere, right, and we're pulling those sister chromatids apart. And again, the sister chromatids are genetically identical. So the whole point of this, right, is that each of these potential daughter cells is going to have one complete copy, identical copy of every gene in this individual's genome, right? And so now we sort of refer to them as daughter chromosomes rather than sister chromatids, which is kind of confusing, to be honest. So we're shortening the microtubules, and that sort of ratcheting action moves the chromosomes towards the centrioles, towards the centromere, and the microtubules that are not attached via kinetochores to the chromosomes are essentially like actually growing longer, and they're sort of pushing against each other. And this whole thing basically causes, one, the chromosomes to move, right? And they're moving towards the poles. And two, the whole cell kind of becomes elongated, and that's where we sort of the spindle becomes sort of even more football shaped as the cell itself gets stretched, really. So in anaphase, we have separating of the chromosomes. Okay, so we've pulled the what were sister chromatids, right? Now are chromosomes on their own, right? We've pulled them apart in anaphase. And so now in telophase, as you can imagine, we're starting to sort of reverse the process. Now that we've made the actual division of the genetic material, we want to reverse the process so that we can corral our new genetic material while we start dividing all the other parts of the cell, right? So one of the first things that happens in telophase is the nuclear envelope starts to reform. And I've got a video coming up that's a short, really nice animation actually of the process of mitosis, showing the dance of the chromosomes and all that good stuff. It does mention that we don't quite understand the process of how the nuclear envelope dissolves and reforms. That's not entirely correct. It's an older video. And we now do understand that it actually has to do with phosphorylation of our lipid bilayer components. And so in dissolving, there are enzymes involved in basically dephosphorylating and causing the nuclear envelope to start kind of falling apart. And then we have enzymes that reverse that process and essentially put that nuclear envelope back together, link up those component pieces of the lipid bilayer, such that we now have that double membrane outside. That's maybe 2 layers, right, of the nuclear envelope that is forming around our daughter chromosomes. The chromatin itself starts to basically decondense, right? It starts to kind of unravel and unwind and go from being this nice condensed linear formation to something that's sort of more loose and kind of squiggly. And we're going to talk, when we talk about gene expression, what the reason for that is, right? If your chromatin is packed and it's wound really tightly with its histones and packed really tightly together, we talked earlier that it's much easier to move it, right, without necessarily breaking off pieces or leaving things behind, right? What we want is genetically identical daughter cells that have copies of every chromosome. All the information in our DNA needs to make it into both of those daughter cells. And if we try and do that when the chromatin is not condensed, then we're likely to actually have broken chromosomes or missing pieces of information. Again, it's much easier to move something when it's all packed together one place rather than having it spread out everywhere. However, when it's all packed together in one place, we can't read those instructions, right? So you don't actually have too much, if any, protein synthesis happening during mitosis because your DNA is basically packed away. It's not available to be read and translated into protein at that point. It's sort of like, okay, the ribosomes are going to have nothing to do for a while because we're actually in the process of dividing the cell. We've copied everything, and now we're actually going to divide the genetic material, and then we're going to divide the cell itself. And we're going to end up with daughter cells that have all their pieces intact and are ready to go. So telophase is basically the reverse of prophase. We're decondensing the chromatin. The nuclear envelope has reformed, and as we'll notice, here it says cleavage furrow, right? And a it's not an arrow, but basically a line indicating where that's happening. Those are the microfilaments now, not microtubules, but you'll notice that the spindle is also, the microtubules are gone, right? They have basically been broken down, and they're all contained within that centrosome. So we still have our centrosomes with their centrioles, right? But the actual spindle fibers, the aster, is gone. Those have basically been broken down. You don't want to destroy them, right? But they broke them down into pieces, and then you can use those pieces for other things. So the microfilaments, though, now are coming into play, right? Those are those actin and myosin fibers that we typically see in muscles. But in animal cells, we use them for cytokinesis, right? This is the process of essentially dividing the cytosol, dividing the cell from 1 into 2. And so in animals, we essentially pinch off, using those microfilaments, we pinch off one cell from the other. It's very much like drawing a purse string. And those filaments basically become tighter and tighter with each other until eventually we bud off one cell from another. So telophase, we're reversing the actions of prophase, and we've started basically, halfway through telophase, cytokinesis has already started.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating 2 new cells in this ongoing cycle that sustains all life.
Okay, so as we saw in the video, right, you can actually watch those fibers, those actin and myosin fibers in the microfilaments form the cleavage furrow and bud off one cell from another, right? So this is how cytokinesis happens in animal cells. In plant cells, you actually don't have that happening at all, but you can see in the diagram here at the bottom, we have our nucleus, right? Basically, they go through telophase, right? So you have decondensed the chromatin, and the nuclear envelope has reformed, and all the bits have been copied. And basically, instead of pinching off one cell from another, they lay down a new cell wall, right? Because remember, animal cells don't have a cell wall, but plants do. And so in between the new daughter cells, different parts of the cytoskeleton are laying down the foundation for that new cell wall, right? That's composed mostly of cellulose fibers, right, which are nice and stiff, and they're packing those cellulose fibers into what's called the cell plate. And that basically gets extended to where it merges with the cell walls of what was the original cell, and you have essentially just subdivided that cell, right? So instead of pinching them off and they become two separate cells, you essentially lay down a subdivision within them. It's almost like subdividing your apartment or something like that, right? If you had a studio apartment and you lay down a line, that's down, one side to the other and divides the apartment in half. That that new line, right, becomes the new cell wall that separates the daughter cells, and then the daughter cells start to kind of expand and get a little bit bigger. And so that's just a difference between plant cells and animal cells. In one you have the cleavage furrow, which is driven by the microfilaments, and in the other you have the formation of the cell plate that's of course going to form the new cell wall composed primarily of cellulose that's going to connect the other cell wall pieces to form two smaller daughter cells. Okay, and finally, just a quick overview of how this process happens in prokaryotes, right? So eukaryotes use mitosis, also meiosis, as we're going to talk about in class, to form spores or gametes. But prokaryotes are already single-celled. And so they too, if they want to divide, right, if they want to reproduce, right, they reproduce asexually by essentially copying themselves and then splitting apart. So it's a really similar overall process to cytokinesis, but because their chromosome is circular, they don't do mitosis, right? They don't have the dance of the chromosomes and all of that, right? Because their chromosome is basically one big loop. And so what they do is a process that's called rolling circle replication. And we'll talk about this a little bit more when we talk about DNA replication after spring break. But for now, essentially they are going to copy their DNA in a similar manner to how eukaryotes do it, but of course a little bit different because they have a circular chromosome rather than a linear one. They're going to copy that, and now I've got two copies, and then the copies basically move along with all the other parts of their cell, which again, they don't have organelles, right? There's ribosomes, and they have cytosol, and there's all these enzymes and things, but they don't have separate organelles. So they don't have the cytoskeleton filaments and the tubules that eukaryotes have. So essentially, the origins essentially grow in opposite directions, as you can see, sort of down here. Let me see if I can get my little thing right. So right here, right, which I've done a really lousy job. Let me make that a little bit better. Right in this spot, I essentially have my two cells are growing apart. So one's moving that direction, and the other one is essentially moving that direction, most likely with the help of their cilia and a few other parts of their bodies, and moving apart. And they basically stretch the membrane. It's almost like if you had any folks who like gummy worms or gummy bears, right? They're sort of that gelatinous material. And if you pull them apart, right, they get thinner and thinner and thinner, and eventually they snap, right? So this is the same kind of idea. You're pulling those two cells apart until eventually there's sort of like so much stress on the lipid bilayer and their cell wall, which they do have a cell wall, that it essentially snaps apart, and you form two daughter cells. And so this is the whole process that we call binary fission. So again, prokaryotes do not use mitosis because they don't have any of the component pieces for that. They instead will copy their genome and then essentially grow apart to the point where you have the formation of two daughter cells.